Hello.
Hello, everyone.
Hi.
Hello.
Good morning.
Again. It's 12:30, but not many people have joined yet. I'll wait a little bit longer. Okay, several things I want to do today. First of all, we'll have a quiz on Wednesday, and I'll describe that in detail in a little bit when we go over some of the questions that you've submitted. And speaking of questions, I'd like to do that first, to share my screen. And you should be able to see the Word document, which starts with questions. So what I did, this was an assignment, and quite a number of you submitted issues that you wanted to have discussed in more detail. And so I have them here prioritized. The most frequent question was about how you deal with a mutation in a DNA sequence. And so write this down. This is going to be the only question on the quiz on Wednesday. The only question that I will ask you is to do one of these problems. So I'll give you a DNA sequence, and I'll say this is the wild type. And then I'll give you another DNA sequence, and I'll say this is the mutant. And the sequences will be different from each other. Then I will ask ask you to transcribe the wild type sequence from DNA to RNA. And then I will ask you to translate the RNA sequence to a peptide sequence. And finally, I will ask, what type of mutation is it? So that will be the only question that will be on the quiz on Wednesday. So let me go through this to make sure that you understand how to do it. We'll take this mutation example 1. So the original DNA strand, so I'll give you one. It won't be this one, but I'll give you one comparable to this. And a mutation occurs. So this T in the wild type becomes an A in the mutant. So the question is, what is the effect on the peptide? And then there was another example, again, the same original DNA sequence, but a different mutation occurs. This G becomes an A. What is the effect on the peptide? And so I've worked it out here in this summary. So if we look at the wild type, here's the sequence of wild type. And if you transcribe that, as you know, T in RNA becomes an A. A becomes a U in RNA. Remember, there are no T's in RNA. C becomes a G, A, U, G, C, T, A, and so on. So that is the transcription of the wild type sequence. And then if we look,
there would never be a T in RNA.
yeah, your name, please.
Yolanda, you don't know how Yolanda did.
Okay, Yolanda. All right, I just
Yes.
have to record this. Yeah. There are no T's in RNA. So whenever you're transcribing from DNA to RNA, wherever you see an A, you would immediately think T, but in RNA, it's not a T, it's always a U. So there are no T's in RNA.
I have a quick question.
Yeah.
You know how you gave us the like the peptide worksheet to tell us? Yeah, will we always be given that, or do we have to memorize it?
You mean this codon table?
Yeah.
Yeah. No, I'll always give you a codon table to use.
Okay.
So the quiz on Wednesday will have included in it a codon table.
All right. Thank you.
So speaking of the codon table, let me make it a little bit bigger. If we look at the codons in RNA of wild type, we take the first codon, AUG, and go to the codon table. The first base is A, it's here. Second base is U, that's here. And the third base is G, AUG, and that says methionine. So the first amino acid in this wild type sequence is methionine. Everybody see how we get that? Then we go to the second codon, UCA, and we look at the codon usage table. We go to U, and the second letter is C, that's here. The third letter is A, UCA, that is serine. So then you write down the second amino acid is serine. And you go to the third one, and I won't go through all of these, but the third one specifies asparagine. The third one, cysteine. The fourth one, arginine. And the last one, UGA, is a stop codon. And there were a couple of questions about what is a stop codon. It's a codon just like all the others, except it's a signal for the ribosome to stop translating, to terminate the protein at that point. So UGA, if you look at the codon table, the first letter is U, second is G, third is A, UGA is stop. And you can see that there are several codons that specify stop. UGA is one of them. So then if we go, so that's wild type. If we go to the first mutant sequence, the first codon in DNA is unchanged. It's TAC, same as in the wild type. The second one is different. So wild type is AGT, the mutant is AGA. So when that is transcribed, A becomes U, G becomes C, and A becomes U. So now we want to know what is the peptide sequence. Again, the first codon is the same, so the first amino acid will be methionine. The second one is UCU. So we go to UCU, here's the U, here's the C, here's the U, and that is serine. So we see that the second amino acid is the same in the mutant as it is in the wild type. So this is called a silent mutation. So it is a mutation in that the DNA sequence has changed, but it's silent because the peptide sequence has not changed. So is that clear to everyone? All right, then let's.
So the met, the sear, and those are the effects of the peptides on the RNA and DNA peptide. These are the
That
effects of the
sequence
peptide.
of amino acids is specified by the DNA and RNA sequences. So the point here is that if you change the DNA sequence, it may or may not have an effect on the RNA sequence and may or may not have, well, it will have an effect on the RNA sequence. It may or may not have an effect on the amino acid sequence. If we look at mutant 2, again, we compare the DNA of the wild type up here. The first codon is the same, TAC. The second one is different. So instead of AGT, we have AAT. So when that is transcribed, it goes from UCU to UUA. So in this second mutation, the second codon is UUA. So if we go to the codon table and look at first codon is U, second one is U, and the third one is A, that's leucine. So now when we look at the peptide sequence, first amino acid is methionine, the second one is not serine, it's leucine. But then all the rest of the amino acids are the same because only one codon has been mutated. And this type of mutation is called a missense mutation, which means that a single base pair change causes a change in one amino acid.
Professor, I have a question about that. This is Alicia Carroll. So would that happen in each RNA strand, or would it just be in select random RNA strands? I don't know if that makes sense, but as the RNA breaks off of the DNA because it's relatively shorter, is it happening on every RNA strand, or is it just on some of them?
No, when the RNA is transcribed from DNA, all the RNA molecules will be the same. So if there's
even with mutations.
Yeah, the mutation is faithfully maintained in RNA.
Okay, thank you.
Okay, anything else? I'll come back and talk about this a little more when we finish up the slide set later today. But for right now, I just want to make sure that you all understand it up to this point, because as I said on the quiz on Wednesday, this is the only thing I will ask you. To take a DNA sequence, transcribe it and translate it, and then tell me what type of mutation occurred. Well, there were a couple of questions about intron splicing and the skipping of introns and exons. So let me just review again what we went over in class earlier. What you see here on the left is a DNA strand, and you can see that there are dark blue bands called exons, red bands called introns. So the introns always occupy the space between two exons. And when this DNA is transcribed to RNA, it's called a pre-RNA, and the exons are included along with the introns. So the entire thing is transcribed, and then the next step is that the introns are spliced out. So all the red bands are gone, and you end up with a mature mRNA which is what is then translated into protein. You see the protein here. And then, of course, the protein is modified post-translationally into a tertiary structure, which is the active part. So this is the process. And I've circled here something I'd like you to remember, and that is that only eukaryotes have introns. So splicing occurs only in eukaryotes, and it occurs only in the nucleus. And you remember that only eukaryotes have nuclei. And so that's the only place that splicing can occur in the nucleus, because nuclei are unique to eukaryotes. And then there was a question about alternative intron splicing. And that's shown in this diagram, where you have several different options that introns have. So if you look at the top here, it says full splicing. You have 3 exons, 1, 2, and 3 in color, and 2 introns separating them. Now, if both of these introns are spliced, so that means that there's a splice site here and another one here, here, and here. You look at the mature RNA, it has no introns, and it has all 3 exons, 1, 2, and 3. Now, an exon can be skipped. So if there's just a splice at the end of the first exon and another splice at the beginning of the 3rd exon, then the entire 2nd exon, along with the introns, are gone. So what you end up with is just exon 1 and exon 3, and that is the coding sequence. Now, there can be alternative splicing at the 5' end. So the first intron would be spliced out. For the 2nd intron, there could be a splice site at the beginning of the 3rd one, and another one in the middle of the 2nd exon, so that what you end up with is a complete exon 1, just the 5' end of exon 2, and the full exon 3. And the opposite of that can also happen here in alternative 4, where the splice site is at the where the 3' section of exon 2 is retained, and the 2nd exon is spliced out.
You're saying it could take just parts of it, it doesn't have to take the whole thing?
That's right.
And that's in the alternative only?
Yeah.
Okay.
Well, that's one alternative. That part of exon 2 can be included, but part of it is spliced out, along with.
They're the only ones that would take part of it and not the whole thing?
That is an option that introns have when splicing occurs. One option is that part of one exon is lost along with the intron. And it doesn't have to be the same part. So in alternative 4 here, it's the 3' end that is saved, so that you end up with a full exon 1, 3' end of exon 2, and all of exon 3.
Professor, this is Steve Stenson.
Uh-huh.
So what you're saying is that according to what is sent out or what is written in via the DNA, it can transcribe whatever it feels, whatever is necessary according to what is written.
Yeah,
And those choices are made by where the introns are and are not.
Yeah, and we're not going to go into what causes these choices. I just want you to be aware that the choices occur. So go back to here, splicing occurs. And in this example, the full intron is spliced out in each case. And what I'm saying is that that isn't always the case. Sometimes all the introns are spliced out. Sometimes they're not. So, and in fact, this last case here, an intron remains. One intron is spliced out, the other intron remains, and that intron then becomes part of the coding region of the gene.
So then, Professor, this Nicholas Gonzalez, essentially, this is just different ways of the splicing process that has a different outcome. Is that essentially what this is right here?
Yeah, it's the reason I mention this is that it's a means of gene regulation. It introduces variation into the genome because some introns may be spliced, some may not be spliced. Parts of exons may be spliced out. So starting with the DNA sequence, you may end up with many different sequences of RNA with the same gene.
Professor, I have a
All right,
question.
thank you.
Okay.
My name is Priscilla. Do we have to remember all of them for the test and how they work?
For the quiz on Wednesday, no, this will not be included there. I might on a subsequent test ask you something about intron splicing, but I'd just like you to know the general principle that introns, first of all, introns are spliced, and secondly, they may or may not be spliced in any given RNA. So introns have options. Okay, let me go on. There were a number of questions about the causes of mutations. And there was one question asking why the more mutations, the greater the consequences in the protein. And I'll try to deal with both of these together. So first of all, what causes mutations? The most common cause is replication errors or copying, when DNA is copied. And I'll remind you a bit more of this in a little bit. There are chemicals that can damage DNA, radiation, and I'll give you an example of how radiation can damage DNA. And then there is genome editing, which is a new method that I hope we have time to go into today. But if not, we'll do it next time. Okay, well, so let's take mutation caused by ultraviolet radiation. If you remember, we talked about thymine dimers, also known as pyrimidine dimers. So UV light, if there are two thymines on a DNA strand adjacent to each other, then UV light can cause these thymines to fuse, to form covalent bonds between them. So on the right here, you see that illustrated. Two thymines form a thymine dimer, and that causes a bend in the DNA. Here it's referred to as a kink. Think of this kink as just a bend in the DNA. And now I'd like to remind you of how complicated copying is and why this kink could cause a mutation. So to remind you of that, let's go back. You remember the video where the guy goes through all these 6 different enzymes that are involved in DNA replication. Now, this is only the copying of DNA. It has nothing to do with RNA. This is only DNA replication. The main enzyme I've circled here in red, DNA polymerase 3, that's the enzyme that adds, that does the copying. It takes a template strand and adds the corresponding nucleotides to it. But it needs all these other enzymes in order to be able to do that. So it's a complicated process. The DNA polymerase goes along adding bases, adding bases, adding bases. It's supposing there is a bend in the DNA, and the DNA polymerase comes along, and instead of adding the correct base, it may add an incorrect one, or it may skip a base, or it may skip several bases. All of these would be copying errors. They would all be mutations. And then to make it even more complicated, remember this happens on the leading strand, and there is a lagging strand where you get Okazaki fragments. And that involves two more enzymes, RNase H and DNA ligase. So to do this lagging strand, new primers, RNA primers, have to be put down so that DNA polymerase can start and do some copying in this direction. And then when it bumps into a primer that's already there, RNase H removes that. And then DNA ligase seals the gap. It's called a nick, the gap between them. So I mention Okazaki fragments because it's a real problem for DNA copying, and it's another source of mistakes, copying errors. And every copying error is a mutation. So if we go back to here, where you have a thymine dimer causing a kink in the DNA, when all of this machinery, all of these enzymes are operating on copying, this kink can cause mistakes. There was, going back to this question, where was it? Why, when there are more mutations, there are greater consequences? Well, there are a lot of different examples of one mutation versus 2 or 3 or 4. I use this example of Lou Gehrig's disease, amyotrophic lateral sclerosis, which is lethal. And what happens is there's a mutation on chromosome 21. So the first of all, here is a healthy person's DNA. You have the DNA sequence, all of these codons. In the case of Lou Gehrig's disease, there's a deletion of this C. So if you delete one base pair, what happens? It's called a frame shift. And so for now, in the patient with this mutation, the second codon, instead of being CGC, take the C out, and it's now GCC. This G, this C, and this C. And the next codon, instead of being CTC, it's TCG, and so on. Every codon downstream of this deletion of a cytosine, every codon is different. And so this entire protein, after the first 3 amino acids, it codes for a completely different set of amino acids. And the consequence of that is huge. it ends up with the death of the patient. So the point I'm trying to make here is responding to why are more mutations likely to have greater consequences, because they change the amino acid sequence more, and that is likely to have a greater effect on the physiological job that protein does. So it states here that after the mutation, every other base moves one place, which makes all the amino acids incorrect after that. Okay.
I have a question.
Yeah.
So there is like any specific reason for that deletion?
No. Mutations in general are random occurrences. And most often they're copying errors. So I've just gone over why copying errors occur. Sometimes they can be induced to occur more frequently, but in general, mutations are random. So a person who gets this disease is just unlucky enough to have a deletion of this cytosine at that position.
Or as you said before, maybe it's genetic, like because of the father or the mother, they have like some kind of gene that made that deletion?
No. I think as far as I know, this disease is not inherited. It occurs as a result of a random mutation that just happens to be this cytosine at this position.
Same thing with cystic fibrosis?
Yes, same thing for cystic fibrosis, and you'll deal a lot more with this when I assign you your mutant.
So Professor, when you have the genes for the was it cytosine, the CYTOSINE for ALS, then you say it's not genetic. So it's something that happens later in life. Is that something that your body just it's like medicine or something you missing or something that makes it do that?
It's a
If you're
mutation.
not born with it?
Yeah, it's a mutation that occurs during the life of the patient. So the patient starts out being healthy, undergoes this mutation, and it changes this protein such that then the symptoms of ALS start. And it's a sort of a slow process, but it ends up with the patient dying.
Oh wow.
Professor, Alex Ellis here. I was gonna ask, a quiz 4, is it this Wednesday?
Yes, this is the one where I'll ask you to do transcription and translation.
What sort of material is it based on? Is it based on today's slide and just today's slide?
Yeah, and I'll talk more about it when we come back to the slides, but it will be this process where I'll give you a wild type sequence, I'll give you a mutant sequence, and I'll ask you to tell me what's the sequence of the transcript and what's the sequence of the amino acid, of the resulting amino acid. And then I'll ask you, what type of mutation is it? And I'll come back to types of mutations later today.
Okay.
There were a number of questions about CFTR. One was the process of going from the gene to the protein to the membrane, and also what is the job of complex sugars that are attached to CFTR. So the job of the sugars, they have two functions. One is most proteins are not active until sugars are attached. And that includes CFTR protein. And the other function of the sugars is to direct the protein to the proper cellular location. So this happens in the Golgi. The protein is glycosylated. Sugars are added, both core sugars and complex sugars. And the composition of those sugars tells the protein where to go. So in the case of CFTR, it's targeted to the plasma membrane. It doesn't go to any other membrane. It doesn't go to the mitochondria. It doesn't go to the chloroplast. It doesn't go to the vacuole. It only goes to the plasma membrane. So think of the Golgi as like a mailbox or a mail sorting room where all the mail comes in and the addresses are read. The addresses are the sugars, and then they're sent off to their proper locations. Why do CFTR mutations happen? Well, we just talked about that. They're random. Differences between mutant and wild type. Someone asked about the purpose of the regulatory domain. So if you remember, we look at the structure of the CFTR protein. It has these transmembrane domains, which form a channel through the membrane. Then there are nucleotide binding domains, 2 of them, and these are involved in providing energy for chloride ion transport. It's an active process. So, and for every chloride ion, there's an ATP that's required to send it through the membrane. And then there is this regulatory domain. And someone asked, okay, what does this do? Well, the regulatory domain is like a switch, and it's mediated by phosphorylation. Well, this is really going a bit beyond what we will talk about in this course. But since someone asked, I thought I would give you a brief look at what a phosphorylation dephosphorylation switch is. It turns out that the phosphorylation is done by an enzyme called a kinase, and the dephosphorylation is done by another enzyme called a phosphatase. So if you look at this little green blob here, this is, let's say that it's an enzyme that has been activated because a certain amino acid, serine, has been phosphorylated. In other words, a phosphate group has been attached to this serine, so that this serine and this arginine can interact, and that holds this tertiary structure in a particular position. Now, if a phosphatase comes along and removes this phosphate group from serine, now serine and arginine no longer interact. The enzyme changes shape and it's no longer active. So the regulatory domain that you see here is this switch. So the CFTR protein has to be phosphorylated in order to be active. And if a phosphatase dephosphorylates it, then that particular molecule in the membrane would no longer be active. And so it really is a function of turning the channel on or off. If it's off, it collapses, nothing can go through it. If it's on, it transports chloride ions. So that is, I think, explains what all the domains are for. The transmembrane domains provide a channel. The nucleotide binding domains provide energy. The regulatory domain determines whether the channel is active or not active. Okay, another question that we'll go into a lot more next time. Someone asked, how does the inability to transport chloride ions lead to the symptoms of the disease, cystic fibrosis? Well, I have a diagram here which attempts to explain it. So we have a healthy section and a cystic fibrosis section. If the CFTR is working properly, it's transporting chloride ions from the inside of the cell to the outside, across the plasma membrane. And when that happens, water goes along with it by a different mechanism, but water is also transported, which makes the surface of the cell hydrated. Surface of the cell has a mucus layer, and if there's plenty of chloride and plenty of water, everything is happy. That there can be a flow. Any bacteria that are suspended are swept away, and the cilia on the epithelial cells are free to do their jobs. Now, what happens if there's a mutation in CFTR is that there's no chloride transport, there's no water transport, and this layer, hydrated layer, is no longer hydrated. It's dehydrated and becomes thick and sticky. The bacteria can't move. There's no flow. The cilia can't sweep things away. And so what happens, and this disease is most prominent in, for example, the lungs get clogged up with mucus. Similarly, it happens in the digestive system, in the pancreas. The pancreatic ducts, which produce digestive enzymes, get clogged up. They can't produce the enzymes, and what enzymes are produced can't move. And we'll talk a lot more about this next time. But in answer to this question that someone posed, the consequence of CFTR mutation is mucus buildup. And the reason is there's not enough water on the cell surface. Someone asked about what is the cure for cystic fibrosis? Well, this is part of your mutant project. You'll investigate this when we get to that. And there were several questions about the core, the sugars, and where the glycosylation happens in this journey from the gene to the membrane. So one of the questions was, where does where are the sugars, polysaccharides, produced? Well, they're produced in two different membrane systems. One is the endoplasmic reticulum, that's where the ribosomes are, and the other is the Golgi apparatus. And each of these is a membrane-bound structure. And so if you look at this diagram, here's a membrane, a phospholipid bilayer, but it's not the plasmid membrane. It's one of these internal membranes, either in the endoplasmic reticulum or the Golgi. And so sugars are anchored to these membranes by the structure called dolichol phosphate. And they can branch, and then they can be attached to the polypeptide, in this case, CFTR. And this would represent, in the endoplasmic reticulum, would represent a ribosome in the membrane. So sugars are assembled in these intracellular membranes and then attached to the protein in the same location. So if you go back to this passage, you have the CFTR gene and a chromosome. It's transcribed into RNA. The RNA goes into the cytoplasm where it is translated, and the core sugars are attached across this membrane, and then it moves to the Golgi apparatus where the complex sugars are added. And as I said, the complex sugars are what tell it to go to the membrane, which it does by passage through a series of vesicles. Now, the mutations that can occur in CFTR can cause effects at any of these different stages. So a class 1 mutation results in no protein at all. Class 2 mutations occur in have an effect either in the endoplasmic reticulum or the Golgi. Then there are class 3, 4, and 5 mutations. And finally, class 6 mutations where the protein gets to the membrane, but it either does not function at all or it functions only partially, doesn't transport chloride effectively. And this is another example. This is in your slide set showing the 6 different classes of CFTR mutations. So this is normal. This is with no mutation. You get transcription, translation, glycosylation in the endoplasmic reticulum and in the Golgi, and you end up with fully functional chloride transporter. Class 1 mutations, there's no protein. Class 2 mutations, there is protein, but it's destroyed, it's misfolded, and it's destroyed before it ever gets to the membrane. Class 3, you get a protein, it ends up in the membrane, but it's defective. And class 4, 5, and 6 are similar. They're all cases where the protein is made, it goes to the membrane, but it's defective. And when you're assigned a mutation, it will be one of these 6 types. There was a question about the chromosomal location of genes, this code. And I show you, once again, you've seen this before. This is the chromosomal location of CFTR gene. It's on chromosome 7. So in the code, the first indication is 7. That means it's on chromosome 7. The next is the letter Q. There are 2 arms for each chromosome. The short arm is called P. The long arm is called Q. So CFTR is on the long arm. So the second symbol here is Q. The third is the number 3, and that refers to this cytological band at the end of the chromosome. 1 refers to a subband, and 0.2 refers to a subband of the subband. And so this has been done with every chromosome in humans and many other organisms as well. And this tries to make it a little more explicit. If you start with the DNA sequence here at the bottom, so that's the sequence. On the physical map, it's the number of base pairs from one marker to another. This is called a cytogenetic map, which means it's based on looking in a microscope at the structure of a chromosome. And so I've said, for example, supposing this is chromosome 9, then if the code was 9p 11.31, 9 means it's chromosome 9, P, it means it's a short arm. 11.31 would be this blue band right here. And 9q, 11.2, 9 is the chromosome, Q is the long arm. 11 would be, 11.2 would be this gray band. And just another example, this is human chromosome 14. And again, the codes would be 14p11.2. That would be this band. 14, that's on the short arm. Q is a long arm, 13.3 would be this white band. And 14q32, again on a long arm, 32.32 would be this gray band way down here at the bottom. So that's how the code works. It just designates at a gross level where on a chromosome a gene is located. And finally, there was a question about peptide bond formation. So I'll just go over it from the beginning. Every amino acid has two functional groups, an amino group, which is always NH2, and a carboxyl group, which is always C double bond O, OH. In a generic amino acid, you have the two functional groups and a side chain, which is variable, and that's what determines the 20 different amino acids. Now, to form a peptide bond, is what's called dehydration synthesis occurs. Dehydration means a loss of water. So in this case, we have a molecule of glycine, which has this structure, and a carboxyl at one end, next to an alanine, which has this structure, a methyl. And in dehydration synthesis, water comes out. So this hydroxyl and one of these H's, it makes H2O, that goes off as water. And this bond becomes the peptide bond, because now instead of attaching to this hydroxyl, it attaches to this nitrogen. And you see that here. This carbon is this carbon. This nitrogen is this nitrogen. This bond is the peptide bond. The peptide bond is defined by its context. So if it's in between a C double bond O and an NH, it's a peptide bond, and that means it's a protein. If it's any other context, so here, for example, is a bond between a C and an N, but it's not the right context, it's not a peptide bond. Similarly, this bond is between a C and an N, but it's not a peptide bond. This is the only one that is a peptide bond.
I got a quick question.
Yeah.
So I noticed for the peptide bonds, in a lot of ways that we learned, it has like the oxygen to the double bond to the C, and then it has the line over and then the N, but typically the H that's going down is upwards. Does that really matter where the position of that H is attached to that N?
No, it could be up.
Okay, so it's just kind of arbitrary depending on how you want to describe it, or what's kind of like easiest for you.
Yeah.
Okay, cool. Thank you.
Okay, I think that's deals with the questions that you posed in the assignment. Is there anything that that anyone asked that I haven't dealt with? Okay, then I will stop sharing this. I want to finish the slide set from, from last time. Do you see the slide? CFTR protein?
Yes.
Okay. We went through this slide set by and large last time, and we ended up with a couple of points I want to make here. So this is, again, back to the question that I'll ask you in the quiz. You have to take a DNA sequence, a wild type and a mutant sequence, and transcribe them and translate them, and then tell me what type of mutation it is. And that's the point of this slide. There are various different types of mutation. A point mutation is just one base pair change. And all of the mutations in this table are point mutations. Then there are duplications, deletions, inversions, insertions, and translocations. You should know what these are. And I think, yes, I've given you an illustration of each of those. What a duplication is, it's just a piece of DNA is copied and repeated. A deletion means that a piece of DNA drops out, it's just missing. An inversion means a piece of DNA is flipped over. So the sequence is reversed. It goes from, in wild type, green, orange, blue to green, blue, orange. A translocation means that two chromosomes exchange parts. So in this case, chromosome 4 changes one of its ends with the end of chromosome 20. So you end up with these two translocated chromosomes. And then finally, there are insertions. An insertion can be as little as one base pair, or it can be several base pairs, or it can be a huge chunk of DNA. It could be many kilobases of DNA. It can all be insertions. But the important point for you and for addressing the quiz on Wednesday, if there is an insertion of one base pair, it causes a frame shift, so that all the amino acids downstream of that are different. So make sure you know what a frame shift is and what the consequence of a frame shift is. And then the effects of mutations, a silent mutation we talked about. So there is a mutation in the DNA. There's no change in the protein. So it's silent, has no effect on the protein. A missense mutation, one of the examples was a missense mutation where there's one base pair change. It does alter the protein, A frame shift, as I've just said, it's the addition or loss of one base pair, and it changes the reading frame so that everything from the mutation on downstream, all the amino acids are different. And that, in fact, is what happens in ALS, Lou Gehrig's disease. There's a cytosine missing, changes the frame, all the amino acids are different, and it's a lethal mutation. Then a nonsense mutation, remember the word nonsense, it's one base pair change, which results in a stop codon. So the translation stops prematurely. You get a truncated protein that is usually not active at all. Okay, CFTR mutations. There are over 2,500 different mutations in the CFTR gene. Most of the mutations are either single base pair substitutions or small deletions, like one base pair or 2 or 3 base pairs. There's the most common mutation is called F508del. That means that a phenylalanine residue at position 508 is deleted. And that causes a change in the protein, so it no longer transports chloride ions properly, and you get cystic fibrosis. Not all of the 2,500 mutations actually result in symptoms. Only about 400 do. And in the group that we'll deal with, I think it's about a dozen, all of which end up with symptoms. This is just to make the point that a mutation, a CFTR mutation, can occur on one chromosome, but remember that every cell has 2 chromosomes. So it can occur on one chromosome only, or it can occur on both chromosomes, the same location. There may be mutation in the CFTR protein, and then there may be a different mutation on the homologous chromosome, not CFTR, but another gene. This other mutation could occur instead of on the companion chromosome, it could occur on the same chromosome as the CFTR mutation. So these are all issues that you'll deal with in your project. Do you have 2 mutations or 1 mutation, 1 on each chromosome? Now we've already gone over this, so I won't go over it again. Following the protein from the gene through the ribosomes, the Golgi apparatus for sugar additions, through vesicles, and finally to the membrane, where wild type makes a normal channel. The mutant proteins that get to the membrane are defective in their transport of chloride ions. And this, you should take a look at when you can consider it at length and in depth, and look at what happens starting with a normal person, where you have the nucleus, transcription, translation, the endoplasmic reticulum, a nascent CFTR protein that's decorated with sugars in the Golgi, and it ends up fully functional in the membrane. Class 1 mutations have no protein at all. Class 2 mutations make a protein that never makes it to the membrane. Classes 3 through 6, the protein gets to the membrane, but in every case, there's something wrong with it. And as I keep saying, the mutation that you're assigned will be one of these 6 types. And finally, this is your study guide for this slide set. I haven't talked yet about, let's see, yeah, I haven't talked yet about COVID and SARS. So you can ignore those questions for the time being. But think about the other questions that I've posed. Now, what I wanted to do was review the answers to test 1, but we're almost out of time. And so I don't want to start that until I have time to complete it. So in addition to having a quiz on Wednesday, I'll review the answers to test 1 and then go on to talk about cystic fibrosis.
Professor,
Uh-huh.
kind of a weird question. It doesn't have much to do, like it has to do with mutations, but I didn't know when to ask it, but I've been meaning to. So cancer is a mutation, right?
Yes,
What type of mutation is that?
it could be any type of the things we've talked about. It could be a point mutation, it could be a deletion, could be an insertion, a large insertion or a small insertion. Anything that causes a change in the sequence of the DNA could conceivably cause cancer. Now, I should say that cancer, you're right, it is a genetic disease. You only get cancer if you have mutations, but they have to be mutations in just particular genes, and there have to be several of them. So to be a cancer cell is really a pretty tough life, because you have to, there are genes called oncogenes, which if you get a mutation in an oncogene, you're very likely to get cancer. If you have a corresponding what's called a tumor suppressor gene, then even if you get a mutation in an oncogene, a tumor suppressor gene will help correct that. So cancer, it is a genetic disease, but it's a very complicated genetic disease.
Okay, that makes more sense. Thank you.
Okay, anything else?